Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Hello everyone. This is a new video about how to use a microscope and how to prepare a wet mind slide. So in order to use the microscope, step number one, you need to know the parts of the microscope. So first of all, you need to know that this part is called the objective lens. This one is the rotating disc, which has, this one is the eyepiece, the eyepiece lens. This one is the, right here is the rotating disc that has the objective lens. The objective lenses are basically in different magnification power. The smallest one can magnify up to 4 times. So if you use it and use this one, that means you are magnifying 40 times. If you are using this one, which is 10 times, and use this one, that means you are magnifying 100 times. The 40x all the way with the objective makes it 400 times. So basically in your examination, you are going to use only this one, which is 10x as well as 40x. You are not supposed to use the 4x or the 100x in the examination. So those are the lenses. Now about the stage. So this part right here is called the stage. Now it is the place where you place your slide. And right here you're going to see the clamp. So what you need in order to fix the slide is basically to pull the clamp and then to place this slide. Okay? So in order to do that, you need to make sure that the objective lens is at the smallest one, which is 4x, and you need to lower the stage to the bottom. And in order to do that, you simply use the coarse adjustment. So this disc right here, this node right here is called the coarse adjustment, while this one right here is called the fine adjustment. Coarse adjustment and the fine adjustment. So in order to use the microscope, step number one, you put the stage to the bottom. Okay? So simply, this part right here is this light source, and the light source has a condenser which condenses light at the specimen. Right here underneath the stage, you're going to see something called the diaphragm. Now, this diaphragm or shutter that basically controls the amount of light that passes through the lens. After knowing all of those things, you need now to know how to use the microscope in order to view your specimen. So let's first prepare our wet mount using onion, and then we're gonna see how to see the specimen using the microscope. Okay. Pause. So this is how you can make a wet mount for onion epidermal cells. Now, wet mount, from its name, it should be a wet specimen. So you can use only water to make your specimen wet, hydrated all the time. Or you can use another kind of solution such as iodine. So if you use iodine, that's going to make it easier for you to observe cells under the microscope. Now if you don't use dyes, if you don't use stains, simply it's going to be harder for you to observe cells under the microscope. Okay, now step number one, you need to place only 1 drop of water at the top of your slide. Okay, and then you're going to get your epidermal cells. In order to do that, simply you can crack the onion and then use your forceps. Use the forceps in order to get a piece of the epidermal. Okay, this one is small. It's gonna be, sometimes it's gonna be hard, sometimes it's not, it depends. So this one is easier. So simply you can take your forceps and you take piece of the epidermal cells. Now if you have a plate, then you can simply cut the epidermal cells. Now we can hold the epidermal layer with the forceps gently, not to fold the specimen. Folding the specimen is gonna make it somehow not clear under the microscope. So you make sure to make it hydrated. As I said, you can use iodine or you can use methylene blue. Now this dye right here, the blue one, is methylene blue. You can use also iodine. So methylene blue stains it blue. After you do so, you can get simply the cover step. And the cover step is this small square of glass. Okay? Now you use it to not to damage the lenses of the microscope itself. You don't need to, it's very thin. So make sure you take only one. Now after you do so, you hold it from the sides and you go like 45 angle or degrees, and simply you drop it, not to trap any air bubble inside. After you do so, you need to Step number 1, turn on your microscope. Step number 2, lower the stage to the bottom. Do not ever forget that. And then place your specimen at the top of the source of light. So for example, some students goes like this. Now the light's going through the glass, so you see nothing basically. Make sure that the light goes through the specimen. And after that, using the smallest objective lens and the coarse adjustment, keeps raising the stage while you are looking through the microscope until you see the field of view. Now field of view basically is once you see the cells. Now if you are a beginner or you don't know how to use the microscope, it's going to be hard for you to judge what is the field of view. Actually, it's going to be also hard to show you what is the field of view using this technique. It's better to use, there are some microscopes that has camera fixed on them, so it's going to screen the view on the board. So I don't know if you can, guys, using the mobile camera in order to show the students the field of view. Can you come closer? Let's see if you can. I think, yeah, somehow like that. So basically, you're gonna see cells. Let's forget about that. Let's forget. So here's the field of view. Okay. Now, once you find the field of view, you need to go through the second lens, and in order to do that, simply rotate this disc. And the only one you use is the fine adjustment. Do not ever use the coarse adjustment again. It is only the fine. While you are looking, you're gonna sharpen the image and you're gonna see the field of view. You're gonna see cells, larger cells, on larger magnification. Now, in order to make it further more magnified, you can use the other lens, which is 40x, okay, and use again the fine adjustment in order to have large cells. Okay, guys. Now you have an eyepiece graticule, okay, which is basically an eyepiece that has scale inside. You can use this scale to measure the sizes of cells. Now it is your teacher's part to show you guys how to use the eyepiece graticule in order to measure the length or the width of the cells in your field of view. Okay, guys. Now it's your turn to prepare the onion slide. And once you finish that, you need to measure the mean length of cells. That means you're going to take 5 cells and measure the length of 5 of those cells and calculate the mean of them. Okay? Thank you.